as we go along, if you have a question or if you're having some kind of problem, let me know by raising your hand or yelling out. Many problems, like the person next to you might be able to help, either looking and seeing what they're doing, or they might help, because there are 35 or 40 of us here. How many?
28.
28. So, I mean, some problem. I mean, try to help each other to some extent. But if you're having trouble, let me know. How many of you are new to QTL mapping? And how many of you are new to R? How many of you just learned R yesterday? Okay. You're at my talk yesterday. I mean, the main idea in QTL analysis is that in the experiments that we're going to focus on, you can start with two different inbred strains and you cross them. The simplest cross you can do is a back cross, where you take the F1 hybrid and cross it back to one of the parents. Then these back cross offspring will all get one chromosome that's from one parent, and another chromosome that's recombinant. That could be the pink chromosome or the blue chromosome intact, but mostly it's a mosaic of those blue and pink chromosomes. And then we would get phenotype data, a quantitative trait measurement on each of these animals or plants. And we would have genetic marker information where, at sites along the chromosome, we know what genotype there is. And if we, the simplest analysis is to look at each marker and see whether, is there an association between whether you got the pink or the blue allele from that F1 parent. Is that associated with your trait value? The backcross is the simplest one to think about, because every individual has one of two genotypes. They're either homozygous or they're heterozygous. And so looking for an association between genotype and phenotype, you do a t-test of, are those two groups, they differ in mean? More commonly, one will do an intercross, where you take two F1 siblings and mate them, or I guess in plants, some plants take one F1 individual and self it. And then at any one position on a chromosome, you have one of three possible genotypes. The data we'll have for from such an experiment would be, we'll have a bunch of offspring from across of that form, and we'll have phenotype data from them. I'm going to use as an example data on a mouse intercross with about 150 animals, I think. And there are a number of phenotypes here. I'm showing body weight and heart weight. That they weighed the mouse and they cut it open and removed its heart and weighed the heart. And here's a scatter plot of heart weight and body weight that they're vaguely associated. This data, all the mice are males. So that the first piece of information is on all of these individuals we'll have phenotype information of some kind. The second bit is we have a genetic map of some set of markers across the genome, that on each chromosome we have some number of sites along the chromosome where we get to observe genotype. And the third component of the data is the genotypes themselves, and at each of those markers along the chromosomes we have, and for each individual, indication of, was it homozygous blue, where it's marked blue here, homozygous pink, where it's marked pink here, or the green is heterozygous. And then there'll be some set of missing genotypes, where you add a marker that was genotyped there'll be individuals whose genotype was not observed. And that's the dark blue kind of background. Usually there'll be some markers that are missing more data than others. And our goals in the QTL analysis, the reason that we did this experiment, are first to try to identify QTL, quantitative trait loci, regions of the genome that are contributing to variation in the quantitative trait. And second, potentially also to identify interactions among QTL, of whether the effect of one locus depends on genotype at some other site. We'll get back to that again later. Kind of a second goal would be, if we've identified a locus as having an effect on the trait, giving an interval where we think the kind of causal gene actually sits. So where we estimate the QTL to be won't be where it really is. The estimate will be with some error. And we want an interval to say how close do we think we are to, what is our understanding of kind of the precision to which we have localized that QTL. And thirdly, we want to estimate the effect of the QTL. That say, if we took a blue mouse and put one little bit of pink in it, at that QTL, how much would the average phenotype change? In the work that I do in QTL mapping and biomedical research, we're principally interested in this first goal, just identifying the loci. And I think getting good estimates of the locations of those QTL is really secondary. If you can't identify the loci, then there's not much value in trying to say where they are. And the estimated effects of the QTL, while sometimes important, is really very far down on the list of importance. I think in plant breeding, the value attached to those different goals would be quite different. That the effect of a region is maybe more important than whether you're really sure that it is a QTL or not. Getting a sense of, or exactly where it sits, perhaps, that you're going to end up taking, a chunk of a chromosome and introgressing it into another line, and having a good understanding of what effect that might have would be maybe more important than actually trying to find the gene underneath. So, I think always keep in mind that there may be some difference between the way that I think about the problem and what I think is important, and rather than what's important to you in plant breeding. And the statistical structure of this problem, as I see it, is that we have some set of phenotypes that are affected by some QTL that we want to discover. And there may be also other covariates involved. That could be environment, or in mice, the sex of the mouse, or some treatment effect. And a main issue that we have is we don't get to observe QTL genotype. That we see genotype only at these discrete markers along chromosomes. Well, we don't get to see QTL genotype, we just get to see these marker genotypes. The markers and the QTL genotypes are associated because of linkage. They're sitting next to each other. A QTL will be sitting next to a marker and it'll have very similar genotypes. The closer it is to that marker, the more similar the genotypes are. The markers will have some apparent association, some association with the phenotype that's indirect and is due to it being associated with some QTL that's causing some change in the phenotype. And the QTL mapping problem, I like to split into two parts. One is what I call the missing data problem, that we get to observe genotype just at these markers, and they're not likely causal on having a direct effect on the trait. But we want to know about genotype at these QTL positions. And so using the marker data to try to impute genotype on the rest of the chromosome. This part of the problem is well solved and well understood and is quickly disappearing in that it probably won't be too long, I think, within my lifetime, that it'll be cheaper to just sequence an animal or sequence a plant rather than try to, genotype some previously discovered set of markers. And so it won't be too long where we will know genotype everywhere in the genome for every individual. But for now, for most of my work, I get to see markers that are, say, 2,000 markers across the genome. And 10 years ago, I would typically see 50 markers across the mouse genome. And I wish I had 100. So, but the second part of the problem is the more important one. I call it the model selection problem. Is that, imagine that you knew genotype at every polymorphism between those initial parental strains. You want to figure out which of those sites in the genome actually matter in affecting the phenotype, and how those, discrete set of QTL positions and the covariates are combined to produce the phenotype. And that problem is much harder. But let's switch over and do a bit of R code and then come back to that question again in a minute. So we have the QTL package loaded and QTL charts package loaded. Now we want to load some data. To load, the data set for one of these experiments will have to include all those phenotypes. The phenotype for all individuals, the genotypes at a set of genetic markers, and then information about the locations of the markers in the genome. There are a variety of, the function to read in data into RQTL is called read.cross. It reads data in a variety of different formats. The one that I mostly use is just an especially organized comma delimited format. The first argument to this function, the first thing you say is the format of the data you're reading in. So we're going to say CSV for comma separated value. The second argument is a directory. We can actually we can grab data directly from the web. So we're gonna grab a data set directly from the rqtl.org website. So we can put as the directory http://rqtl.org and double quotes around it. The next thing we need to give it is the file name. The file name is sug.csv. SUG is short for Sugiyama, who's the investigator that did this experiment. Note that, so if I type a bit of R code and press return, and I didn't give it a complete line of R code, this prompt for me will switch to a plus. That indicates I need to keep typing. So I give it the format of the data set, the directory in which it occurs, which here we're just going to grab it directly from the RQTL website, the name of the file, and then there are a ton of other arguments. The most important one specifies the way in which the genotype data were encoded. for this particular data file, this is an intercross, and the genotypes were coded CC, CB, and BB for one homozygote, the heterozygote, and the other homozygote. So we need to tell a recross function that genotypes equals C parentheses. This bit of code right here is creating a vector with 3 values, these 3 character strings. Another argument that's useful is to give it some allele codes. Just tell it that store information about a single letter code for the 2 alleles in the cross. We give this basic to show up in plots that have genotype letters. It will use those 2 letters to code the 2 alleles. If you don't put alleles equals C, 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 B, it will just use A and B as the 2 alleles, which often leads to confusion later. And a parentheses to end it, and we all press return at the same time. And it should say, read the following data, 155 individuals, 93 markers, 6 phenotypes, and it inferred from the data that it was an F2 intercross. The most common problems that one has in doing this sort of thing is the set of commas that are in between each of these arguments, and the C parentheses around these. We're creating vectors of character strings. And probably the most common difficulty one has is specifying the directory for the file. That if you have the file on your hard drive already, of figuring out how to tell R where that file is sitting. But that's part of the advantage to us here of just grabbing it from the web. Has that worked for people? If you're having problems, raise your hand. So recross grabs this file and uses this information to interpret it and reorganizes the genotype, the genetic map, and the phenotype data in some complicated internal structure that I'm happy to explain later if you want, but we'll leave as it just makes some object. This left arrow type thing, the less than sign and the dash, that is assigning the result of that function to an object, and giving that object a name. So it should be in your environment here. You should now have Sue as an object in your workspace. If you type ls parentheses to list the objects in your workspace, you should see Sue sitting there. If you type summary Sue, so this is a rather complicated object because it has these 3 rather disparate sets of data organized in a complicated way. If you type summary Sue, this summary function knows the type of object it is and will give you a summary appropriate to that type. So if I type summary Sue, it'll tell me it's an intercross, 155 individuals, there are 6 phenotypes. And this is the percent phenotype data for each of the 6 phenotypes. It tells me I have 19 chromosomes and they're all autosomes. There are 93 total markers, and this is the number of markers on each chromosome. And 98% genotype data, and just over all the markers, 24% CC, 50% CB, and 26% BB. This function also does a bunch of other checks to make sure that there's not something weird about the data. Yeah.
We have an issue here. She got some, when she loaded the QTL package, she got something that says detach, and none of the rest of us got that. And so now it's not loading her file. Detach package QTL? We didn't get that.
That maybe happened if you clicked on it over here. If you unclicked on it. Just type library and then yeah.
Okay, now you can get this double up. That'll work for your.
Yeah, so when you click on a package over here, it will just do library and then that package name over there. Deselect the package over there, it will that detached thing is like the opposite of library. Remove it. There are a bunch of little functions that you can use to pull off little bits of information to remind you about, to figure out about the cross structure. And in for the number of individuals. MAR, or total markers, just tells you the total number of markers in the cross. And MAR, just the number of markers on each chromosome. It'll tell you how many markers are on each chromosome. The number of phenotypes. You can get the chromosome names. The phenotype names. There are a number of little functions like that to just pick off little bits of information about the process. So in addition to, yeah.
So when you're looking at, when it reads the raw data, how does it know, like if we have to create our raw data set, do we have to do it in a specific way so it knows how to read it?
Yes. We'll leave that to later.
Okay.
That part proves the most painful in most cases. Painful for you to get it in the right format, and painful for me to make sure that I'm not reading, garbage, and that I'm giving you informative errors if it doesn't conform to my specification. Let's leave that to later.
Okay.
So if I type summary SUG, it gave me that little summary. If I type plot SUG, it'll give me a summary plot that includes a plot with the pattern of missing genotype data. It gives me a plot of the genetic map, and then it gives me a histogram for each of the phenotypes. And if there are categorical variables, like sex in this case, it just takes the value 1 for all individuals and give you 1 big bar. And a number of those pieces you can also get separately. If I type plot missing, it'll just give me the pattern of missing genotype data. So for each individual and each marker across the genome, just a black dot to indicate that that individual is not observed for that genotype. If I type plot map, it'll give me just the plot of the genetic map.